Hello, everyone. Welcome to the Thomas Expert Series. Thank you for joining us today. We're going to get started in just a minute. If everyone could just double check, make sure their microphones are muted. This talk will be on demand within 24 hours, so please let your colleagues know how much you enjoyed the presentation today and have them sign up for future talks or direct them to the on-demand tab. I'm curious where everyone is tuning in from today. Rob, where are you at?
Rob, can you hear me, Heather?
Yes.
Rob is in Manchester in the UK.
And then we've got Florida, Mississippi, New Jersey, New York, Philadelphia, Massachusetts, DC, North Carolina. So a lot of East Coast. And then we've got some California, Utah. Awesome. All right, we'll go ahead and get started. At this time, I'm going to turn it over to Rob here to talk about raising the standards in bioactive protein manufacturing and innovation.
Thanks, Heather. Much appreciated. Good morning, afternoon, everyone. My name is Rob Nixon. I'm Commercial Director here at QKine, and as Heather said, here to talk about raising the benchmark for bioactive protein manufacturing, recombinant growth factors for stem cell culture. So we'll dive straight in. So what we'll cover today, our mission at QKine, the value proposition that we offer to yours and our customers, where you can get hold of a lot of resource and deep dive a little bit on the product portfolio and some of the applications that we help support customers with. So who are we? We're based in beautiful Cambridge, UK, founded in 2016, so we're relatively young. And we are focused on manufacturing these highly expensive, complicated recombinant proteins. And our genesis came from one of our co-founders who was an expert in the TGF-beta family, and he was providing from his lab these proteins to the Cambridge Stem Cell Institute. And in a chance meeting with Catherine Elton, our CEO, formerly from Abcam, they determined there was a company to be found. So 8 years later, we're now serving 37 countries with our customer base, and we're very much a scientist-led run company. So our reason for being is to raise the benchmark and standard for these proteins. There's a lot of variability, there's a lot of private labeling, there's a lot of unknowns in the supply chain when it comes to these recombinant proteins. There's also been a distinct lack of innovation in this space, and I'll come to that later, but we're here to help support some of these emerging fields, like, organoid technology has been around for, maybe 10 years now. There's obviously a drive towards cell therapy, with a lot of biotech spin-outs from various different university hospitals and biotechs and pharma companies. And we're here to disrupt the supply chain of growth factors, in your case in North America. So one of the problems that is inherent in this market that we're still astonished by today is the low quality proteins that are in the supply chain today. And you can see from this little infographic, there's a lot of variability coming from another commercial supplier, and that lot variability inherently means that customers have a risk that their cell culture won't work. And you can imagine that if you spend 90 days culturing some neurons, you better hope that your cell culture works. So what we've done is really try to solve this by really honing down on the manufacturing, the quality control, really rigorous testing, around purity, bioactivity, to ensure that real consistent, reproducible product goes out of the manufacturing. All products sterile, mycoplasma-free, bioburden-free, and new standards for endotoxin levels, low endotoxin levels as well. So we're really serious about our commitment to making sure that when a customer switches to us, there's inherently less variability than buying a second lot from their existing supplier, which makes that switching customers a lot easier. And the second step in this is that a lot of the products that are manufactured today are still using HEK or CHO cells, and inherently that leads to contamination from animal-derived components. There can also be other growth factors contaminating the vial, and there's a drive by the FDA to remove any animal-derived components and remove that any sort of risk. So again, we've resolved that through making sure that there is absolutely zero animal components in any of our manufacturing. In fact, we've done it to the tertiary level, so there's absolutely zero risk of cross contamination. And the next step to that is it means that we've been able to produce some of the only commercially available animal-free products like HGF, TGF, beta 1, and DKK1, which I'll speak about later. And the third step is, bringing innovation to this space. There really hasn't been very much. And, apart from anything else, these proteins with poor bioactivity really means that it drives cost for customers. And it also means that this is a critical bottleneck, in terms of COGS for cell therapy developers. So to strip that out, we need to bring that innovation to the market. So, I'll speak a little bit later about our thermostable FGF2. But there's several other examples on our website, and we're in the stage of a portfolio build for the time being, but you'll definitely see over the next sort of 6 to 12 months some really innovative technology and products coming to market from Qkine. But bottom line, we want to make the most active product for customers that's more stable, improve that reproducibility in cell culture, and ultimately give customers the best possible opportunity for the best possible outcome for their stem cells. So we have this sort of 9-point quality commitment for customers, and it really encapsulates everything that I spoke about. So, this animal origin free certified facility. We just completed our ISO 9001 recertification that's enhanced for anyone who wants to head to that sort of preclinical space. Really accurate protein recovery from vials, so customers know exactly what's in the tin. Fully quantitative bioactivity analysis, with guaranteed lot reproducibility, really setting those new standards for protein purity levels. And, ultimately, we want to get customers the best quality product. So, those 9 sort of critical points are what we're all about, really, to make sure customers have the best possible product. So why are they so important, growth factors? Well, they're really essential reagents in driving stem cell fate. And you can see from this infographic, I know it's quite small, and you can go on our website and you'll see the infographic in much better detail. But this sequence of growth factors determines what a cell will become ultimately. And that's why it's interesting for us as a business, customers using quite a number of these products, but also, it means that we have to have a breadth of products for our portfolio as well. So, customers can really buy, no matter what they're working on, they can really buy, sort of from a one-stop shop, from an expert in manufacturing these proteins. So again, transparency is something that we're big on. You can see everything on our website. Products are listed by cell type, research area. There's application notes. There's technical notes. Publications are something that for sure, researchers and academics are asking all the time, and we're getting more and more peer-reviewed papers. And that, again, there's links to protocols on the website where products have been used in their processes, blogs, you name it. It's all on the website for you and your customers to tackle. And of course, if there's any questions that you have, you can reach us anytime at support@qkine.com. So just a little insight into some of the application notes and tech notes that are available there. So we started off as a company that was focused very much on iPSCs, stem cell maintenance and expansion. As we've grown up as a company, we moved into that organoid space, people doing 3D cell culture, wanting to do drug screening, disease, toxicity modeling, whatever. And then we've moved towards the neuronal set of stem cell growth factors, very complex, difficult to manufacture, and now we have a complete set there. We've also seen this trend over the last 3 years, people interested in the cultivated meat industry. So we filled a bit of a hole with some species-specific growth factors, where customers are really interested in non-human growth factors. And then, sort of by design, it's now led us to the more hematopoietic stem cell space. So people who are working in T cells, NK cells, that area of research. So that's, really exciting for anyone who's, thinking about sort of cell therapy customers. And again, we're really focused on now bringing some of those innovations into this portfolio, engineering these proteins to really drive these different market segments forward. So in terms of the top selling proteins that we have, Activin A, FGF2, TGF beta 1, these are sort of bread and butter used every day
Rob,
by stems.
uh, I'm Anjali. I have a question before we go further. You
Sure.
said it's animal free. So
Yeah.
what is the host? I'm perplexed. Like,
It's E. coli is the vector,
okay.
bacteria we use. Everything is
Okay.
done in E. coli, Anjali.
Okay, because we're talking about stem cells and stuff, and then we're saying animal free. Okay, thank you.
Yeah, no problem.
Okay.
So, Activin A, FGF2, TGF beta, these products used for stem cell maintenance and differentiation, sort of bread and butter of any iPSC core. Some of the really interesting ones in organoid culture, definitely R-spondin 1, which I'll speak about shortly. Noggin, probably my favorite protein, purely based on its name. BMP4, very difficult protein to manufacture at scale, and FGF7 as well. And then for customers in the neural space, BDNF, GDNF, also very difficult proteins to make well consistently. And then the hematopoietic range, we're still building this range out, but we have immediately CSF, GCSF, GM-CSF, and then we just launched IL-2 and IL-7 in the recent months, and you'll see some really interesting interleukins coming out towards the end of the year. And then we have our first foray into extracellular matrices components. So vitronectin is something that's used very widely by iPSC customers. So watch this space. It's quite a complex landscape when it comes to ECMs, but vitronectin is available immediately and widely used. So I spoke a little bit about innovation, so I'm going to move more towards product and the portfolio. So we now have this thermostable FGF2 in our portfolio. So it's used daily. It's an unstable cell culture recombinant protein. It's got a half-life of about 12 hours. We have a license to be able to manufacture the thermostable version, which means that customers are allowed to can basically have weekend-free stem cell culture. So absolutely ideal from a work-life balance perspective for customers, but it's also better for the customers' stem cells because it has a much more consistent signaling of FGF2. And we just had this paper towards the end of the summer. Alessandro Botero's lab in Turin, qualifying and validating our G3 FGF2, and he showed that there was an 8-fold reduction in the need of FGF2 using the thermostable mutant instead. And that's great from a cost perspective because it allows him to really reduce his costs, meaning that he can produce that many more iPSCs. And again, we're living in a sustainable world now, with one eye on supply chain and sustainability attributes. So again, helping to contribute to doing things better, smarter, and less risk to the planet. So jumping to organoids. People doing a vast array of different organoids, wanting to recreate, more human like disease models and systems. So we have a vast array of organoid supporting growth factors. I'm not going to list them all here, but rest assured we can cover basically everyone and anyone who's working with organoid stem cell culture. There's a vast array of products there for customers to have a little look at, and we'll speak about R-spondin and BMP4 now. So BMP4 is really a critical one. It's used in many research areas, especially cardiac, neural and kidney. It's really a difficult protein to make well, and we've really validated it and benchmarked against a number of different manufacturers commercially available today. And bottom line, we have probably the best BMP in terms of bioactivity on the market today. So really exciting protein to be able to offer to your customers. And it's a very expensive one as well, by the way. So, if you want to use it, you want to use a good one. This is a study that was done some years ago at Cold Spring Harbor Lab in Long Island. Our sponding ones used really for conditioning media for customers to generate things like pancreatic organoids. And conditioned media is inherently filled with contaminants and very variable and has very, very poor reproducibility. So we now offer the recombinant version to condition customers' media, which means that they can generate lots and lots of consistent pancreatic organoids, as was this case by the Tuvison lamb at Cold Spring Harbor. So some nice images there, but yeah, a very widely used recombinant protein there. So move to the neural stem cell set now. I'm not going to go through every single one. You can see there's an awful lot of different recombinant proteins here. And these cell cultures can take anywhere between 40 and 90 days. So they're very, very long culture conditions. So again, customers really want to be buying the best possible quality reagents for these cultures, because you don't want to get to 90 days and your cells are not doing what they're supposed to do. So there's some data here from the Sanger Institute in Cambridge. Very pretty pictures of some astrocytes here. And we have a nice tech note on the website that's coming from DZNE in Leipzig or Dresden, where they were using BDNF and GDNF really to help differentiate their neuronal cell lines to be able to help study neuropsychiatric and degenerative diseases. And this is a relatively new portfolio, as I've said before. There's still some gaps in our portfolio, but we've got probably 70 to 80% of the portfolio today now, which is very nice. Again, anyone who's doing sort of immune cell therapy type applications, watch this space, and this will lead us towards clinical grade products, which we anticipate launching probably by the end of Q1 next year, to really be able to address those clinical applications that you see biotechs having. So because we are animal free, using E. coli, there are a couple of totally unique proteins in our portfolio. HDF is typically a very, very long protein. We've engineered it to be very, very short, just the active NK1 fragment used for hepatocyte generation. TGF-beta1 as well is the only globally animal-free TGF-beta today, and the same with DKK. So we start to cover some really interesting applications where customers really have an appetite to remove those animal-derived components from their portfolio, and ones where you can really offer really unique products to customers. Yeah, so I spoke about it before. DKK1, it's really bioactive and totally uniquely animal origin free, and you can see the vast difference here between the different suppliers, where supplier C has no biological activity with their DKK versus our own. So we're really committed to sustainability at QCoin. I don't know if any of you've seen our packaging, but it comes in sort of very brown envelope, sort of artisanal looking packaging, and it's by design really. We want to use recycled materials to enhance sustainability practices. We want to do the best that we can. All of our products are lyophilized, so there's no need to ship on dry ice anywhere in the world. The tubes themselves, they're recyclable, and we're in the process of working on My Green Lab certification as well. So watch this space for the beginning of next year as well, with us having that sort of ACT label. So I think with that, a picture of the beautiful River Cam and some punting, that's it. Thank you very much. Hope you enjoyed that.
Thank you, Rob. I'm going to share my screen for just a second to show you the QKine kiosk. In here, there is some literature, some web links, including the application notes, the link to the application notes that he's been talking about, and videos in here. You'll also see our on-demand tab and our upcoming talks, so you can register for those or watch them on demand. And I will hand it back to Rob to see if we have any questions in the chat.
Why are animal-free products so important? Yeah, it's a good question. I mean, you know, in the late 90s, the UK was in the news for all the wrong reasons with new variant CJD. And of course, medical devices, at that time came under such scrutiny with, adventitious viruses being transferred and, causing horrible side effects to patients with, surgical blades that were contaminated with prions. So, sort of 25 years later, there are still animal products being used, not least in cell culture, but also for sort of translatable science. And, the FDA has made, it very clear that they want to remove any animal derived components, wherever possible, move to recombinant, move to chemically defined products. So, that's why we're sort of aligning ourselves with regulatory standards moving forward, that we want to offer, 100% animal origin free product to ensure there's that translatability and no inherent risks and no problems or questions asked by, regulators and, customer QA.
All right. Let's see what else we have. What is the impact of lot-to-lot variability of growth factors when used for stem cell culture?
Yeah, it's a great question. I think ultimately it depends on, it comes down to functionality. And, stem cells have a, specific genetic, and phenotype and, activity that customers are looking for. So, if you saw from my picture that there's real wide and varied bioactivity for some of the suppliers that are in the market today with these products, that ultimately customers don't know or can't necessarily trust that the product that they're going to use will end up producing their beautiful microglia 90 days later. So by tightening that lot-to-lot reproducibility, we're just making sure that customers are getting the best possible product for the best possible outcome.
Why has there been such a lack of innovation in the bioactive proteins, and where can Q-kind proteins be especially useful?
Yeah, it's another good question. I can't speak to other suppliers, but I can certainly say that I think first you have to run before you can walk. So you have to be able to produce product and product well, which I think we do. And then the next step is, and I think it's really driven by some of these new industries that are cropping up. So, particularly cell therapy, where, recombinant proteins in media is about 70% of cost of the therapies. In order to be able to make those scalable and accessible for the future, you need innovation to inherently reduce the cogs. And again, from a cellular agriculture company, I don't know how many people are familiar with this market. These people doing cultivated meat. It's the other extreme where these guys are looking to produce a commoditized product at maybe $5 a pound or a kilo of mince. Well, they're using sort of bioprocessing type technologies to do that, which are inherently expensive anyway. So there's going to have to be, innovation to be able to, logarithmically drive those cogs down. So, there's all sorts of interesting technology that can be adapted to be able to do things smarter and better. And I have to say, watch this space. We've got a very active IP portfolio, and lots of interesting products coming from Qkind that, Thomas, you're going to have access to.
Are there products kept in the US?
For the time being, I think we shipped between Monday and Thursday to the US for the time being. I think because we're still a relatively new supplier to Thomas, we don't keep any inventory. But I think that will change as we start to scale the business. And by the way, we do about 20% of our total business today is through customers in the US. And it's also the biggest stem cell market in the world as well. So, it's logical that we probably should have some inventory at a certain point. But the nice thing about our products is, they're the size of a, sort of a matchbox. They're very easy to ship. And because they're animal origin free, there's no real issues with customs. So shipping is very, very easy for us for the time being.
What are some of the more difficult proteins you are proud to have mastered the manufacture of, and why?
This is where I wish Luana had been on the call, but from my perspective, definitely TGF beta 1. This is the only animal-free product in the world and used, extensively by biotechs and research. So, that one in particular is a really, really big one. HGF as well is a great example of us taking a really big protein and shortening it, which allows us to be able to scale and pass on those cost efficiencies to customers. So we're able to drive the COGS down there. And a couple of the interesting proteins that are missing from our portfolio that are going to be coming in the next quarter are definitely things like IL-34, which is a huge one for the hematopoietic stem cell space. And I think BMP4 is another one that we've really mastered the craft of manufacturing as well. Historically, very difficult to produce at scale, and we've really cracked the nut. And I think that's purely R&D, science-led company, really honing our craft and our expertise and know-how.
What product do you have that no one else has?
Ooh. Yeah, that's a good question. Well, the TGF beta, the fact that it's animal free, would be probably the most significant one. Yeah, I spoke about DKK and HGF. And I'm not sure that there's any others that anyone else doesn't have. Just that the products that we have are, we believe, benchmarked better in terms of their bioactivity. So again, consider that, we're in a portfolio build stage. We will have some interesting additional products. The other one that is interesting, and it's a bit more of a niche application, is follistatin resistant activin A, and that is interesting for cultivated meat companies, but it's also interesting for iPSC maintenance as well. So there's a core group of iPSC researchers that would be interested in that one.
Okay, I apologize on my pronunciation of this one, but I would expect more difficulty in managing lot-to-lot variability of animal/eukaryotic proteins.
Eukaryotic proteins.
Thank you. In a prokaryotic host.
Yeah.
Also about post-traditional modifications of these proteins, which happens in eukaryotic host.
Yeah, I can sort of bat that one back and come back at a later date, but I have to say that we've really cracked the nut when it comes to production of these proteins and the bioactivity. And I can't really speak to the differences between eukaryotic and prokaryotic post-translational modification per se per molecule, but they're recombinant human proteins, and when we benchmark against competitive, even CHO or HEC derived ones, we don't necessarily see that there's any issue using E. coli to produce these products.
That's good to hear that.
But we'll come back to you, Anjali, with a robust R&D science validated to answer, if that's okay.
No, that's okay, because it's like, for me that goes without saying when you're expressing these kind of animal proteins into E. coli. That's why you stay away from it. That's why most people stay away from it. But I understand that for in vitro research, that has to be done to manage cost and the whole workflow, which is so tedious when you work with stem cell lines. So I understand the need for it, but at the same time, you won't get not like everything which you would generally get when you have the eukaryotic host as a, expression cell line. So there's always like you lose some, you win some. So
yeah, exactly. Yeah. Good. Any other questions, guys?
Yes, there is one more. What level of genetic drift do you see growing growth factors in a bacteria versus a human or a yeast cell line?
Great question. Not one for the sales guy, I don't think. So Isaac will have to follow up with you separately, if that's okay.
Sounds good. Any other last minute questions, guys? Alrighty. Thank you so much for joining. We do have 2 more talks this year. We have Streamline Your Projects, why Eldon James is your go-to toolbox on November 15th, and Consolidating Hand Protection for Growing the CE Market on December 6th. See you guys then, and thank you, and have a great day.
Thanks, Rob. Have a great
Cheers,
weekend.
Heather. Thanks
Thank
all.
you.
Have a
Bye.
good weekend.
Thank you, Rob.
Welcome.